Hi, my name is Mohamed Baljevic, and I'm Associate Professor of Medicine at Vanderbilt University Medical Center. I'm a Director of our Multiple Myeloma program and Director of our Amyloidosis multidisciplinary programs as well. I've had the privilege of being on an abstract which I think is sharing some interesting and important data in terms of ABBV-383, or otherwise recently more known as Etentamig, new name for this bispecific antibody. So ABBV-383 or Etentamig is a BCMA-binding bispecific antibody which has already presented its phase 1 and published its phase 1 data in terms of efficacy and safety. What we're going to showcase in this ASH meeting is phase 1b data of a step-up dosing study that was done subsequently in order to optimize the delivery of this bispecific antibody in a step-up dosing fashion, which is very convenient. There's only one step-up dose followed by monthly delivery. So every 4-weekly delivery of this bispecific antibody, which is fantastic. No other bispecific antibody that is currently labeled has this type of step-up or dosing schedule from the very beginning. So what this study did is it employed several different step-up doses in terms of the Etentamig dose as well as the Dexamethasone pre-medication. The dose that was taken forward was a schedule of 2 milligram of bispecific followed by, or pre-medicated by rather, 10 milligrams of Dexamethasone, followed by, on day 4 of cycle 1, 60 milligram full 4-weekly dosing that was pre-medicated with 36 milligrams of Dexamethasone. And then on day 1 of each subsequent cycle, Etentamig or ABBV-383 was given every 4 weeks. So what this led to is a really impressive low CRS overall rate of 30%. So all grade CRS only 30%, of which 26% was grade 1 and 4% was grade 2. No grade 3 or higher CRS events. So this is really impressive because no other bispecific antibody has demonstrated this low rate of overall CRS without the aids of other medications beyond dexamethasone. Furthermore, the overall response rate didn't seem to change that dramatically from the previously published phase 1 experience. Overall response rate was 69%, and vast majority of that overall response rate was actually VGPR or better, so 65%. So in summary, I think this data is actually the base for registration of phase 3 Trevena trial, which is currently ongoing globally, as well as in the United States and many centers, where ABBV-383 or Etentamig is going to be pitted against a couple of different standard care treatment options in patients who have received at least 2 prior lines of therapy, so 3rd line and onward. And again, this dosing schedule of small dose of 2 milligrams of drug pre-medicated with 10 milligrams of dexamethasone on cycle 1, day 1, followed by a full 60 milligrams Q4 weekly dose on day 4 of cycle 1, pre-medicated with 36 milligrams of dexamethasone, to be taken then, monthly from cycle 2 onward every 4 weekly with a full 60 milligram dose. So that's something that's going to be employed in this phase 3 registrational Trevena trial. So we think this is really important because it showcases both, excellent safety signal as well as maintained efficacy signal. Our patients are frequently frail, have different considerations that makes them ineligible to keep coming to hospital for more frequent dosing schedules, and developing a strategy that delivers bispecific antibodies in a very convenient monthly delivery is surely something that we have seen has already been welcomed by patients and certainly has potential to positively impact availability and use of this particular bispecific antibody in community settings in particular.